Assalamualaikum warahmatullahi wabarakatuh, and hello everyone. My name is IZZATI binti RANI, and I'm going to present my research project. And this is the research aim and also the general objective. Narrow down to the specific objectives, I have 4 specific objectives, and I would like to present the graphical abstract for my current study. Now we come to the results and discussion. First objective is to conjugate daptomycin and PEG and the complexation with liposome for targeted delivery system by using multi-TOF MS and also FTIR, and also using the NMR spectroscopy showed successful conjugation, and also the degree of substitution were reported as 28%. The first step involved in model selection and fitting is to perform ANOVA and select the appropriate type of model. R-square is used as a measure of fit to a model. From the fit statistic summary shown in Table 2, the final reduced quadratic times quadratic model was appropriate to be used to navigate the design space. The adjusted and predicted R-square is in reasonable arguments where the difference between both is less than 0.2. The non-significant lack of fit test is good where it compares the deviation of actual points from the fitted model. The results of the investigation on the parameters used for sample preparation shows that in order to get optimal level of 100 nm of the liposomal size, the sonication time range proposed by the model is at 9 to 11 minutes, while the best ultrasonic intensity is at the regulation range of 70 to 80%. For the polydispersity index, Figure 4 showed that in order to obtain a 0.2 PDI, the best ultrasonic time is at 9 to 11 minutes and the ultrasonic intensity is at 70 to 80%. We move on to the schematic illustration of formulation preparation of targeted liposomes by using dehydration-rehydration method and also preparation of RBC membrane. We use inverted microscope and also TEM, which is a transmission electron microscopy, in order to obtain the morphology of the prepared formulations. As we can see here is the red blood cell membrane under the inverted microscope, and also the ghost RBC. And we also perform the ghost RBC under TEM. We can see here is the formation of pores and also the morphology and uniformity of liposome size produced by the dehydration-rehydration method. We can see the polydispersity of the liposomes, and we can see here is the ring of the liposomes, which have approximate size of 30 to 40 nm. The third objective is to determine the encapsulation efficiency, drug release profile, as well as the efficacy of in vitro testings against MRSA. So these are the formulations produced, which is the blank liposomes, vancomycin liposomes, daptomycin liposomes, and also RBC-coated daptomycin vancomycin liposomes. We can see here is the zeta average. The size keep on increasing with the additional coating of RBC, and also the combination of these two antibiotics, which is the vancomycin and also daptomycin liposomes. They have a good polydispersity index and also zeta potential, and have a 39.76 encapsulation efficiency for vancomycin, whereas 86.65% of encapsulation efficiency for daptomycin. And these are the drug loading efficacy. Red blood cell coating can be seen in the TEM image of the dispersion of RBC-DVL. We can see here the red blood cell coating of the liposomal layer, and also we further confirm by using flow cytometry. In the first picture, this is the liposomal coating of vancomycin, and the second picture is the layer of the ghost RBC coating the resulting liposome, hence triple layer can be seen. All formulations show initial bursts. For example, vancomycin liposomes within the first 4 hours, which is the cleaning phase, releases almost 50% of its content in line with the amount of unencapsulated drugs. Following the cleaning level, all formulations started to release the residual drugs in a steady stream before plateauing. The physical stability of the obtained nanocarriers was further evaluated by examining the morphology changes via TEM and also SDS page. On day 0, it can be seen that the liposomal formulation was much more polydispersed compared to day 14 and above, in which aggregation starting to take place. noted that the presence of protein bands was also clear on day 0 and slowly fades by day 14 till day 21 when compared to the red blood cell membranes, MRBC, most likely due to the leakage of the formulations which eventually rips off the essential protein marker on the surface of the RBC as confirmed by the SDS page analysis. Hemolysis activity was performed to compare all the formulations with the positive control, which is the Triton X, to see the safety of the developed formulation against human red blood cell. In hemolysis study, all formulations produced showed negligible hemolysis activity, which is less than 5%, even when tested at the highest concentration. In the cytotoxicity study, low toxicity was recorded on mammalian cells, HEC293, which is the human embryonic kidney, and also liver cell, which is the HEC-G2, up until 72 hours. This is in good agreement with the result previously reported that the encapsulation of vancomycin inside the liposomes may reduce toxicity caused by the drug. Since the use of a high-dose antibiotics contributes to the death of bacteria, further study needs to be done to investigate the side effects and also the effectiveness of the free drug compared to the entrapped drug. Okay, so this is the cytotoxicity study of the HEC-G2. So for the MRSA study, all formulations showed good result compared to the free form. Last but not least, the fourth objective is to determine the ability of the resulting formulation to avoid macrophage cells and its toxicity rate in vivo model. Incubation of vancomycin liposomes, daptomycin liposomes, and also RBC-DVL reported reduced in macrophage uptake, which is less than 10% when compared to the free drug, suggesting that inhibition of the scavenger receptor-mediated uptake could cause this decreased cellular uptake as reported before. Formulations containing liposomes and also red blood cell coating show increased biodistribution up until 72 hours. The serum biochemical parameters of the treatment group were not significantly different from those of the saline group, and no apparent toxicity was observed after a single dose of the test liposome formulation. In contrast to the free drugs, a slight increase in the serum biochemistry level can be observed in formulations group due to the fact that over time drug will be released. Nevertheless, the rise in the serum level was still lesser than the free form of So for the histopathological evaluations, it can be seen that compared to the positive control, the one with the RBC coating, the formulations showed less lymphocytic infiltration and also tubular dilatations in the kidney and also in the liver. We can see here and we can conclude here that the double coating with liposomes and also the red blood cell coating is said to help in protecting the formulations and also provide a more sustained release delivery of antibiotic as well as enhance efficacy of MRSA eradication. That's all from me. Thank you.